So in general for class tonight, it's going to be a slightly different format than we have normally done in Foundations, where I just talk for an hour and then we do a show and tell. We definitely want to show and tell, but many of us have been struggling with coding or doing other weird things. And so we would like to get to that. But one of the things about tonight is that we have like a real in the room with all of us thing that we're going to do. We're going to do some activities. And so we're going to try and fly through all of biology in a very short amount of time to give you a brief overview of what that is and what sort of strange things can go on inside that green room. And so I've got slides and Nora has slides. And then once we've made it through all of that, then we're going to actually like, you know, swab the inside of your cheek and do some real science. So we'll get to see what a little bit of what your DNA looks like. So I'm going to present more generally about the room and the broad science things. Nora's a little bit more targeted at the activity. And so between the two of us, we're going to have you covered. So if I can find my cursor, we're going to talk about microbiology and biochemistry. We're going to talk about the micropipettes, which are the tools right over there, the blue things. We'll look at the polymerase chain reaction, which is the PCR. Everybody knows about a PCR test from COVID. It's a way that you can make copies of DNA. So we'll talk about that a little bit. Then gel electrophoresis, and then bioleather bioplastics, which is sort of the other camp that lots of things in the bioream can be. And then there's more. So we can go through all of that. First up, we're going to try and cover all of these branches of science in like 4 slides, which is woefully inadequate, but you know, we'll do as best we can. Feel free to correct me if you need to, or know that I've gotten something wrong. So it turns out that you are a whole bunch of things. You think, therefore you are yourself, a person. Presumably we are not all in the matrix and then imaginary players around you, but you never know. But if you want to imagine if you want to believe in the empirical science of it, you are made up of a collection of cells, which have over time decided that they would collaborate in a way that's mutually beneficial. And there's a whole lot of evolutionary detail that goes along with that. But in general, you're made up these little packets. The inside, internally, they carry some information about how they should be built and what they should do. And they've got a lot of the same pieces and parts. There's a nucleus where the information is stored, and that's true whether you're an animal cell or a plant cell. There's these mitochondria, the powerhouse of the cell, you might remember from a high school science class, the thing that actually creates or turns energy that you eat into something that's usable by your cells. And then there's a bunch of other weird like growth things on the side of the nucleus. There's specialized pieces inside of an animal cell. You might have a flagellum so that it can move. You might have other weird things that get attached so that it can move around. A plant cell is going to look a little different. It's got this big vacuole so that it can hold water. It's got all sorts of different adaptations so that they can fill the role that they need to be. There's a lot of the things in your cells that are very similar cell to cell, but you have eyeball cells, you have muscle cells, skin cells, brain cells, all of that. And they're all variations on a similar theme. And so across the board, it's really interesting to take a look at. Also, in addition, science has really taught us in the past couple of years that there's, in addition to all of your cells, a huge part of you is your microbiome. So there's also a bunch of things carried in you that are a part of you, but they do not share your DNA. So like your gut bacteria is a thing that's a part of your body that you need to have to make sure that it all goes well. And somebody who's got that out of whack might feel extra gassy, or any number of things. There's a bunch of different pieces and parts to your biology that's based around these cells and the information and way that they work. It's super complicated, very fascinating, and it's tons and tons of stuff that you could get to know. If you really want to drill down into it, there's biochemistry. So if you're cool with thinking about the cell as a basic unit of how people work, how do you make a little machine that's a cell, and what are its mechanisms? What are the nuts and bolts that hold that together? Fundamentally, it's chemicals. And they're chemicals that are very, very large. The chemicals of your cells are proteins in lots of ways. And those proteins will be chemicals that have many different pieces. This diagram right here is representing individual atoms in a protein. And you can sort of see they have this crazy arrangement. These are other ways to represent that. So here's these spheres are atoms. These are atoms. This is adenosine triphosphate, one of your energy carriers. This is hemoglobin, I think, the stuff in your red blood cells that carries around the oxygen with the iron. And then you've got these other proteins. These sheets and spirals and whatever, they're all coiled up atoms that make literal little chemical machines that are inside of your cells that are fascinating, mesmerizing, and you can find really cool videos, and I'm going to post some in the chat in Foundations afterwards, where you can watch these mechanisms at work. There's some, I think they're in the UK, these researchers that do animations of these proteins and how they mechanically work through your cells and like crawl along fibers as your cells reproduce. There's some fascinating stuff to look at. Really, really cool stuff. Protein modeling is a way that we understand how this fits together. You can take biochemistry classes where you understand the mechanisms of all of this. There's some pieces that are really common to all cells that are alive on Earth, how they process their food, how they turn sugar into fuel. Plant cells will also take light and carbon dioxide and turn that into sugar. So there's some pieces that are specific to some types of cells, and then there's cellular respiration, which is pretty universal for almost everything that's alive on Earth. There's a few rare species of living things that do something different. That's chemosynthesis, that live in like deep ocean vents or inside of volcanoes or other weird places. But most life acts pretty much the same in lots of interesting chemical ways. So just that alone has been a lot. We really, there's a lot to go through here. But in general, cells make up all life. They follow DNA instructions. And what's really fascinating is to see how much of your DNA you share with other things. So your DNA, if you didn't know, you're 99.9% the same as every single other human being, period, the end. You are 0.1% different at the most from any other human at all. So identical. There's almost no difference. A Neanderthal is 99.7. We've killed all of them. Sorry, Neanderthals, or interbred with them. Chimps, 98.8. What gets me is the banana, 60% same as a banana. Because they still have cellular respiration. There's still a lot of the cell that's the same, but 60% of your DNA is shared with them. It's pretty nuts. That like, oh, that's good. The 98% with a pig, 90% with a cat, like just you keep going down the line, 25% with a daffodil. And yeast, like yeast you buy from the store to put into your bread, you share at least a quarter of your DNA with that, which is kind of mind-blowing, right? There's a really, across our planet, in our biosphere, there's a lot of similarity in lots of the different things that are alive. So even though it may feel like some weird niche activity, when you're working in the bioreum, you can be very, very close to some of the deeper hidden secrets of how the world works and operates in ways that are very, very applicable for people. Also, you can take this diversity of DNA and look back into the tree of life. So if you want to see how things are related, it's just how different from them do you look, right? If you think about your own family reunion, you probably look fairly similar to a sibling and a little less similar to your cousin, but you could tell that you're cousins, and then your second cousin or third cousin, it gets a little bit further and more distant. All of that is played out over all of life. And so there's some weird link where you and a fish have a common ancestor, and you can trace back through the DNA and look at the variations and actually read that code if you're very ambitious and try and understand how differentiated you are from them. All of that is something that's definitely within our realm of possibility. Just takes, maximum effort to make it happen. So it's fascinating. 50% with a tree. You're 50% with a tree. So inside your cells, just pointing out the pieces and parts, the nucleus is right here in the middle. Then it's got some other pieces around. The nucleus is really where it can keep your DNA safe and intact. Once the DNA leaves the nucleus of the cell, bad things happen to it. So it needs to be kept inside of a safe container inside of your cells. And then outside of there, you've got these endoplasmic reticulum, you've got the Golgi apparatus, all these different things that we definitely make sure that our 14-year-olds know very, very well so that they can forget it by the time they're 24. With few exceptions. With a few exceptions. There's some people who do really important stuff with that. And I want to say that as I'm being recorded as a science teacher. And Nora definitely makes good use of it on a regular basis. But the majority of people should know that this is the mechanism that helps you work. But the actual specifics is something that, if you're at a dinner party, it's pretty rare to talk to somebody who knows much about the Golgi apparatus. But in any case, lots of these pieces are, even though they might look different cell to cell, there's lots of things that are going to be very, very similar in lots of ways. Inside of a cell, it's a big party with lots of these things pushing up against each other, moving around, jumbling around in space. And the way that they interact is critically important to making you operate, which is kind of nuts. If you wanted to understand how that works, if you wanted to know the programming language of life, it would be the DNA. DNA is a base 4 code system. So there's only 4 letters, G, T, C, and A. Those 4 letters can be arranged in all sorts of different arrangements. And so your DNA is the basis for how all of life works. Now, your DNA is a fairly complicated double strand. So it's a duplicate of itself. That way it has a copy that it carries all the time. It's I think that's RAID 0, where you have 2 copies of your hard drive, right? And so that is a safety measure so that if one goes down, the other's still there. However, life can operate without that. And so RNA was a simpler version that very likely came first. And we still use it back and forth. If you got the mRNA vaccine, your body knows how to read and interpret RNA. mRNA is the way that it sends messages, hence the M in mRNA. It's how your DNA inside your cells sends messages to the parts of the cell to make them do stuff, is with mRNA. And when you got the mRNA vaccine, if you did, hypothetically, or I guess we all did because we're here and vaccinated, it's a qualifier to be in the room. So if you got an mRNA vaccine, basically you're getting a shot into your shoulder that gave your body the instructions of how to build the things that needed to fight off coronavirus. And so it was using the mechanisms that your cells already have. We were just sending it the right message to say, here's how you get prepared. And so it made a whole bunch of the coronavirus spike proteins, and then it learned how to fight them off. It's a really fantastic way to use our own machinery to our advantage. So by understanding it, we can really do a lot. Here's the coronavirus. There's this fantastic New York Times graphic on it and its code. This is the B117 variant, which is not like the beta variant, which was feels like a million years ago. And so they would map out what the individual genetic mutations were. When changes in DNA happen, those are called mutations, and they can be just a single letter that changes. And when a single letter changes, it changes how things are built. It swaps out maybe 5 or 6 atoms in that protein, and so its shape changes. Within proteins, a few changes in atoms can lead to a pretty big difference in shape. And so that's where these mutations have come into place, where lots of the mutations have been in the spike protein that have made the more recent variants more aggressive. So fascinating things there. Although I am not the virus expert, nor definitely is. So if you've got more to chime in, you sure can. But in any case, the DNA and RNA, that's the code of life. That's what we're going to take a deeper look at today. Modifying DNA is something that viruses can do. So this is an interesting piece. You can have a virus that will send in a viral genome into a bacterial genome, and then it will just sort of commandeer that bacteria. This is what happens to you if you get coronavirus, or if a bacteria gets a virus. The virus will inject its DNA, and then the cell will run that DNA code or the RNA code and just operate it like it's its own. It can't really differentiate, and so it just sort of runs the program that it's given. The DNA code in length is sort of along here, so you can see how DNA code changes from material to material, and it's probably too small to see, but mammals have this short genetic code, amphibians have this crazy long genetic code, flowering plants are this very long stripe. And so DNA and code length in terms of the pairs, you can see there's a bunch of variation across life, and it doesn't seem to make any sense to what we consider to be the most complicated forms. Maybe even like backwards, that flowering plants are way more complicated than mammals. Something about that hurts a little as a mammal, but it's fine. It's fine. Not upset about it. And so it's interesting how all of this relates, but that is all of microbiome. Done. We did it. There's so much more that you could possibly learn. It's going to be impossible for us to go through it. But the big takeaways is that you're made up of cells. In the middle of those cells is the code to make it run, which is DNA. And we're going to get access and actually sort of look at that DNA today. We're going to make some copies and see what happens when you have a whole bunch of that all in one space. And part of the tools that you'll need for that is a micropipette. And so we have 4 good ones, 3.5 good ones, one that's a little, that's seen a full life. And so in these tools are the core skill of most of the bio room. There's old school pipettes like this, where you've got a big vacuole here, and then there's like this fun animated GIF of how they work. But really, ultimately, it's a tool that lets you dish out a very specific amount of a liquid. That's it. That's the short version of what it does. It's super precise at giving you exactly the right amount of a liquid, so you can have very controlled amounts and everything can operate exactly as you want. You can have different size tips that would give you different amounts of liquid. They're sort of different lengths. They can be different diameters. There's a host of different differences that you can have. But just understanding this tool is going to be a core part of what we're up to today. The real big thing that we learned last year is don't press it all the way. When you grab, try and stand in front of where the camera can see me. There's this button on top. This is the one button that does this, that does pretty much all the show. You press the button and there's like a place where it gets harder to push. That's the first stop. So when you first do it, you'll press down to there and then let up. And then when you're trying to get rid of it all, then there's another, you can press beyond that. So here, and then go again, and then you let up from there. It doesn't necessarily make sense until you have one in your hand. So don't worry about it. You'll feel it when you get there. But your first press is down to the first stop, and then back up, and then down to the first stop again, then the second stop, and then up. We'll leave this graphic up when it's time so that you can see what that is. But they work with the equipment that's up here. They're just a fascinating tool for dishing out very specific amounts of chemicals. These can be done robotically, so you can absolutely have a CNC machine that does this 100 at a time, which is fascinating. Not something that we have. We're going to do everything by hand, but those machines do exist if you're in a big money lab. But the thing that we're going to do is the polymerase chain reaction. So the PCR test. This is the big trick that we're going to pull today. We're going to take your DNA and we're going to do the first cycle where we separate it apart. Then it's going to add on these little end caps that are going to be places where it starts to copy. And then from there, it'll make a copy of those 2 unzipped pieces of DNA. Your DNA is a double strand that we can unzip and then make copies of each of those unzipped halves. And then it's exponential, so we can then unzip those new pairs. So now we have, instead of 2, we have 4. And you get a doubling and a doubling and a doubling until you have a whole bunch of copies of your DNA. You have, in order to make this work, we need some primers that get the copying started. And then some, oh, we've got fabric in the way, totally in the way. Your DNA will be copied by a polymerase, which is a thing that is able to make a polymer. So it's a protein. ACE is a common suffix for a protein, one of those chemical machines that you've got. And a polymerase is a protein that makes polymers, many part chemicals. So your DNA is a polymer. It's got a whole bunch of little pieces that are all zipped together. And basically, we're going to run this chemical reaction a whole bunch of times. In order to try and make sense of this, here's a video that I'm just going to talk over because it's way easier to see it happen than to have a still graphic. Also, this is done by a brilliant research group. This video deserves a ton of credit, so we're just going to sort of show off really quickly. You'll have some sample, you'll do a DNA and then a target DNA, then we'll do this copying thing. You'll have a whole bunch that we can go through. So the PCR technique is going to be to take this thermal cycler. It's going to help this all go through in the way that it needs to, where you can go through and find some target DNA. When you take your sample, we're going to apply a chemical that'll help you find exactly what you need. And then we're just going to sort of zoom through. All of it's going to happen inside this little teeny tiny pipette, or tiny little tube. So that little tiny tube goes into here, and that's where the magic happens. Inside of there, you'll have these little DNA strands. And basically, what'll happen is it will unzip, it'll find your target DNA. And from there, in cycle 1, it's going to go through and you'll denature it. So you'll take it apart by the denaturing. Just by heating it up, it'll separate out the 2 strands. And so we do this where it heats it up. And then the polymerase, that's the polymerase stuff, it'll sort of hook on to the end of the target DNA. And these primers will get it started. There'll be free extra letters of the DNA, extra segments. And so these attach right at your target point, and then the polymerase can grab on and it finishes up all of that after it loops around and sort of grabs all the letters that it needs to make copies of that part of your DNA. So this is what's going to happen repeatedly as it goes through the denaturization, annealing, and then it needs a little bit more heat to extend and anneal. And so you do this, and the only part that's really actively being copied are these green sections. So that's going to get copied and copied and copied until it overwhelms all the rest of the DNA that's there. And so there'll be some of this red that's copied, but you can see it's not all of it is copied. And you'll get, once you get a couple rounds in, this is just going to be copying those green sections, and you'll have tons and tons and tons of those green sections, and it'll very much overwhelm the rest of it. So you'll be able to see how much of this you have. This is how we could go through and find the DNA of coronavirus in a PCR test, because you can make lots of copies of your DNA in there, and you'd be able to see sort of how much of that you've got. So then after these cycles, this is exponential. So you definitely get a doubling of what goes on. And as we move through these different pieces, you go through a certain number of cycles and your target DNA turns into very, very large amounts. And this fragment is very, very small. You'll notice they're counting in hours. So this is a process that we're going to start tonight, but we won't be able to finish tonight. So there'll be another night throughout the week where you'll have to come back in. We'll be able to button up all the details, or we could probably realistically run it when somebody's here and then close up all the details later on. But that is the short version of what's going to happen. So your actual, the science on this side of what the PCR is going to look like. Also, super big shout out to this team. I want to put them on screen for our video. They crushed it, and now we're just going to get an ad from YouTube. So in here, Canal, I don't want to say that wrong, but they did a brilliant job making that animation. And so once we do all of that, you're going to have a big copy of your DNA from PCR, and then we're going to do gel electrophoresis. Gel electrophoresis is this stuff. If there's a science thing that CSI Miami does to show off what science is, it's the gel electrophoresis that's glowing under a black light. And so it's a real fun looking thing. You can see how your DNA works. Basically, it's a gel filter, and we'll be able to see what kind of gene you have by how much your gel runs. So this is an interesting piece of this puzzle. Basically, you have a collection of your DNA that's going to be put into a series of wells across the top. Here's like 3 traces. I wish I could zoom in on that. Here's this thing. And we apply a big voltage that makes all your DNA want to push its way through the jelly. And as it pushes its way through the jelly, the bigger the DNA piece is, the slower it moves. The smaller it is, the faster it moves. And so we'll be able to tell which DNA variant you had based on whether it's the big one or the small one. Because we do the PCR just the right way, it'll be able to filter that out, and you'll be able to spot, do I have the big gene or the small gene? And that's the resolution that we'll be able to have. So we'll be able to copy your DNA so that we can have a big enough sample that it shows up in this gel electrophoresis. And by running it through, by running everybody's through, you'll be able to compare yours to a known long or short. And the long ones won't move as far, and the short ones will move further across from the positive to the negative, or the negative to the positive. So in any case, I need to get going. Also, we just did a lot. Let's keep going. If it feels too high science for you, one of the cool things that does happen in Barcelona is they make bioleather. So in addition to this, totally different vein, you can save up all your orange peels or coffee grounds and make leather out of it, which is really, really cool. So if you're interested in doing that, there's all sorts of different things that you can do to go along with this. But these biomaterials are really fun things that you can make. You can grow sponges. There's all sorts of cool opportunities within this space. Definitely the sort of thing that you can get a biology degree to understand, or you can find cool instructions and follow along, and then sort of just barely move the needle forward from there. There's really cool stuff, but it's got lots of different varieties. If you want to learn more, the Barcelona Fab Lab is a great place to start. There's a couple other resources online. The Global Community Bio Summit and the Thought Emporium are really good YouTube channels if you wanted to check out some of those things. They've got lots of cool resources to look at. But now it's time to do gel electrophoresis of your own DNA, which brings us to Nora. And so, you ready? Okay.
Oh, yes, definitely. But I did my PhD studying this thing called CRISPR, which is in the news sometimes for genetic engineering. And so I have a few different things that are all kind of genetics-y and bacteria-y in my background. But yes, today we'll not be looking at bacteria, unfortunately, because those are my favorite, but people cells. And so, as you know, and sort of this is stuff we all Corey just went over. It's really awesome. So we all have almost all of us is our cells, like they're all have the same DNA, but then they end up making different stuff like hair and tongue and teeth. But they all have our DNA, except for the bacteria that are on us. And yeah, so today we're going to be getting DNA out of our cheek cells. You could do this with your hair. All of you, you're just constantly shedding cells that have your DNA. That's why CSI works so great. But DNA is one of the chemicals you have. You're made up of a lot of different molecules, and one of them, is DNA. And it's made up of these things. Like each little line on that little strand of DNA, we call a base. And so DNA kind of looks like a little ribbon. And then and so there's all those different bases, and then little segments of that strand are different genes. So they're all. You can tell here, like each different piece of the strand, each gene is like a little different set of bases. And so each of those little bases, those groups of bases and those genes tell your body how to do different things. So even though all of your cells in theory could they have the genes for hair color, even if you're like a tooth, cell, whatever. So they've all got all these genes, but then the one we're looking at today is called PV92. It is not a gene with a known function because there's like some ethical considerations about us looking at your DNA and telling you, like, for example, that you have, some cancer gene or something. I'm not a medical doctor, and unfortunately cannot tell you if you have a cancer gene. But so this one is one that just like everyone has this gene, PV92. Nobody knows what it does, but everybody has a little bit of a different version of PV92. So some people have a version that's 900 bases long, so it's a little bit longer, and some people have a shorter one. And then some people, because you get genes from your mom and dad, you'll have one of each. And so, yeah, so basically you get 2 copies of every gene, 1 copy from your mom and 1 from your dad. And so when we end up looking at everybody's, you'll have some version of these 3 different situations. So a big and a small one, a big one and a big one, or a small one and a small one. But in the end, you can't tell. When we look at them, you'll either have a big one, a small one, or 2 different sizes. And so the way we look at DNA, what we were talking about, it's really hard. It's really tiny. DNA is very small. So it's hard to just like look at it and be like, oh, you have this gene. You can't just like look at it under a microscope. Or if you do, you just see that it's a molecule and you can't actually tell, like, is that a gene for anything? Like, what does it mean? It's impossible. So the way we have to tell about stuff is to like get your cells, we get out the DNA, and then we just like we like have those enzymes, that polymerase that Corey was talking about. We have it like search for exactly PV92 and then make a ton of copies of it. That way, we can actually see without a microscope whether you have PV92 or not, or what size your PV92 is. And so, yeah, basically we'll have this enzyme, the polymerase. It's like a little magnifying glass, kind of like searching for whatever we tell it, which I told it in our little tubes to look for PV92. And then it's going to go and make a ton of copies. And so, yeah, the next thing we do once we've made all these copies is we sort it based on how big the DNA is. And so that's gel electrophoresis. That's the next technique where you like will have this like kind of gelatin thing that you put DNA into. And so DNA has a negative charge. This is just like a fact about DNA. It just happens to, which is kind of handy because when we put it into this gelatin, it will get drawn, through the jelly towards the positive, like how negatively charged things get like pulled towards positive things. And like it'll try to like sift through this jelly, but like bigger DNA will kind of get stuck because it'll be like trying to wiggle its way through this, it's called agarose. And so it ends up sorting stuff based on size, where smaller things like the smaller piece of DNA on the right kind of travels more quickly than the bigger one. And so what it looks like is that this is an example of kind of what we'll end up seeing. And so this is a gel, and from the left, on the left to right is all different little pieces of DNA. And on the left, it's just a comparison for all different sizes of DNA. And so it says base pairs up there at the top, and you can see there's all different numbers. And so at the top would be how big something would, where something would be if it was 1500 or 1200. And so the column next to that one has 2 bands. And so that means that person has the big piece and the small piece. And then the one to the right of that only has that smaller piece. It's like further down. And so that means that person only got the small PV92. And then, you can kind of see there's like, and then that person only got the big version of PV92. So it's kind of just showing the different possibilities for what this will end up looking like. But yes, so DNA analysis is really helpful. It helps us know what our DNA and our genes look like. And then people obviously also use this to look at the viruses that you have inside of you. And so Corey was mentioning, I study viruses. I don't actually study COVID, so I don't have, unfortunately, any like insider info, which, everybody wishes. But I study the viruses that there's like good viruses that live in and around us and like in the environment. And so that's more my shtick, not the bad ones. But yeah, that's DNA analysis is really helpful for that. And what else do I got here? Yeah, just a few more things about the bioroom if anybody's interested. There's we've had a few fun projects in there. We have a microscope. There's cool, if you have anything you want to look at under a microscope. We've done a little bit with fluorescent bacteria, looking at them under the UV. And we also did a class where we built leather out of kombucha, which was fun. Right now I'm helping a member sex her chickens, which is a phrase that I learned through this process, which is when you determine what sex a chicken is. And basically it's really hard to tell by looking at them what genitals they have, basically. And so the best way to do that is to be did it XY or XX? And so she brings me a feather and we're helping to be is this baby chicken? What is its sex? And then we also have some stuff for doing testing your water. And yeah, lots more possibilities. So yeah. Nice. Yeah. Yeah. Is there anybody has any questions about? I'd take a COVID question if I had to. I'm just kidding. Yeah. Oh, sorry. Yeah.
Slightly unmasked, so you can go to the bathroom or whatever you might need to do for that. But I'm gonna stop this recording nonsense, and at this point, Nora, I feel like it's time for you to run the show.